the team for inviting me. Also, I'd like to shout out to my class of 1986. That's my graduating class. I was fortunate enough to have started with the class of 85, so I have two classes to be thankful for. And also, I am very grateful to some of my old teachers, many of whom are no longer with us, especially Prof. Engman, Prof. Bedu, and Prof. Kobe. And then to those of you who are on the call and are from Accra Academy, good bless both the boys and girls. So our theme for today is personalized medicine. And I'll try and keep this as short as possible. It's more to give you a flavor of what the field looks like, what it is and what it's not, and what the future might look like. Okay. So this slide basically illustrates what we do in traditional medicine. Patients come in and we do our best to assess them clinically. We take a history, examine them. You get lab tests as necessary, often guided by what you had and what your examination revealed. Occasionally, we might do some level of imaging. And then we'll put the story together and come up with a diagnosis, again, based on what our history of medicine and what we know about disease pathophysiology teaches us. At the end, we have to choose a mode of treatment. And unfortunately, often the therapies tend to be one size fits all, often driven by cost, accessibility to therapies, and so on. Many of you will acknowledge very readily that it's often trial and error. Okay, so you try an antihypertensive, it doesn't work. You add on more. If it doesn't work, you might change it and so on. So often what we see for our patients is variability in response. This is a major problem in traditional medicine. And indeed, some patients may not respond at all. And not surprisingly, a lot of patients will develop side effects. So this is a traditional approach, okay? So what is personalized medicine? This is the title I was given. So personalized medicine is partially here. I don't want you to believe too much of the hype. To some extent, it works for oncology, but for complex diseases, as I'll show you later on in diabetes and cardiovascular disease and gastroenterology, mental health, and so on, this is an emerging concept, okay? By personalized, we don't mean personalized service or individualized medicine. Again, that's a fallacy because it's not possible to evolve diagnostic procedures and therapies for a single people. That's just not possible. What we're really talking about is subgroup medicine. So my preferred term, and what many people in the field prefer to use, is the term precision medicine. So we're trying to have more precise approaches towards diagnostics based on genomic information, because after all, that's what makes us individuals very different, combined with clinical information and biomarkers beyond what we currently do in order to make specific diagnosis and treatment decisions. So please, if there's nothing to take home from this talk, I'll prefer you use the term precision medicine rather than personalized medicine because the two are not necessarily equivalent. So the panel you have at the bottom is what personalized medicine is supposed to look like. Instead of the individuals being lumped together within a population sense, begin to look at individuals within subgroups so that you have genomic information, clinical information, lab tests, which can be targeted, very sophisticated biomarkers, including proteomics, untargeted, metabolomics, looking at metabolites in body fluids and tissues and so on. A move towards molecular imaging, rather than just taking X-rays or CT scans or MRIs, begin to use molecules to image. So you get more precise information. Once you have all this information and with the appropriate tools, be able to subtype people and come up with appropriate diagnosis, bearing subgroups in mind, And this should then lead, hopefully, to precise therapies, more efficacious therapies, hopefully less side effects. And in the long run, after all, we're very concerned about cost of healthcare. So hopefully make therapies more cost effective. Okay. So for a broad audience like this, I just wanted to throw in some definitions here because this is not something you might be thinking about most of the time. So let's start with the basics, okay? Since in my title I have genome, I'm going to start with genetics. So what's genetics? So broadly, genetics is a field, a study of genes, and genes are the currency of inheritance. That's what makes us similar to our biological parents and yet different, right? Because for every gene, you inherit an allele from your biological mother and a biological father. So in many ways, you are like your parents, but you're not a carbon copy of your parents. You are unique in that regard, and you pass on your traits to your offspring. So keep that in mind. And by genes, we're talking about DNA sequences, part of which would determine protein production. Many of these proteins might be structural, they have other functions, a lot of them are enzymes and so on. And these are tied to disease causation. So we're talking about DNA sequences which code for proteins. What's the genome? The genome is a complete set of DNA. So in human cells, we have about 3 billion DNA base pairs which make up the human genome, right? And the field of genomics, again, is a study or a specialty where you're using genomic information. These DNA, the information encoded in this DNA set for purposes of disease risk prediction. This is huge. You don't want people to develop diseases. It's the predictive power of the field that we are looking for. Our ability to screen, to make diagnosis, and then to have targeted therapies, more precise therapies. And very importantly, hopefully use genomics to also predict prognosis. We're not there yet. Again, don't believe the hype. We're still in the process of getting there. And what is epigenetics? So I've often heard, oh, our genes have been changed for a long time. That's true. The structural genes have been changed, but there's something called epigenetics, where you can have chemical modifications of your genes, which then determine which genes are turned on or turned off and under what circumstances. And there are drivers of these processes, most of which are hormonal. So in my field in endocrinology, and certainly in oncology and other diseases, people are always talking about epigenetics. The other term you may have heard about is metagenomics. So what is a metagenome? We don't live in isolation. So we have our genome, we're surrounded by other living things with their own genomes. And as humans, we coexist with all kinds of organisms, especially bacteria, and viruses, and parasites, and so on. So the metagenome looks at the genomics of all these organisms which coexist with us, especially in our guts and our skin, other organ systems, and do influence our metabolism and the way our body interacts with the environment, and sometimes may drive disease. So please keep these terms in mind as you get interested in the field. Okay. So what are the potential of genomics? So I mentioned earlier on, don't believe too much of the hype, okay? We are getting there. The field of genomics is actually possible now because of 3 key things. One, we've gotten very good at acquiring more biomarkers of disease beyond what you and I did when we were in medical school. And these often come in an unbiased way, big data, and we have to learn how to handle such data, such information. Part of it may be genomics, deep phenotyping, metabolites, which can be screened and so on. At the end of the day, you have to put it together, right? So you need computational power. So again, that's the other major development that has allowed us to move the field forward. And then thirdly, you need connectivity. You need to be able to put things together. So I live in Ellicott City, 14 miles from Baltimore, right? And so my environment is a little different from you who live in Accra. How does that affect who I am as a person, my disease causation, disease risk, and so on? We need connectivity. Not only do you need computational power, you need algorithms which are predictive in terms of disease causation, disease therapies, and so on, right? And we are getting good at that, especially in the field of oncology. So the potential uses are going to be in cancer, infectious diseases. You see part of it with COVID. It's really a teaching moment for all of us, why some people are more predisposed than others, and so on. And in my field, diabetes, which I'll talk about in the next few moments, obesity, mental health, dementia, cardiovascular diseases, and so on. And then especially rare diseases. There are a lot of diseases out there which haven't been diagnosed yet. So this is an opportunity for us to reclassify and name new diseases. Very important in the area of disease risk prediction. This is something we don't talk about, right? In order to prevent disease, you have to be able to assess risk for disease screening, for diagnosis, and for precision therapy, like I mentioned earlier on. And if you are a clinician for patient in front of you, health outcomes for the patient, very important. That's why they see you. It's for no other reason. They want to be healthy and go take care of their family. So they want to know, doc, am I going to get better? So can we use genomic and other tools to be able to predict outcomes? And then for population scientists or for health policy people, you can use this in a population sense, like you're seeing for COVID and other complex diseases. So this is the whole concept. Again, this is your take-home message, that this is going to be the future. Disease risk prediction, screening, diagnosis, and precise therapies. Okay, so I'm not an oncologist, but this is an area where there's been a lot of relative progress. So on the left side of your panel is our current diagnostic approach. You see a patient that you think has cancer. In this case, he has lung cancer. You screen him, you do your imaging, you might get some tissue biopsy, old-fashioned, get your histopathology, and then come up with therapies based on what your old book tells you, right? Many of which are very toxic. A patient might do, or most of them don't do very well, right? And then in the future, we're hoping to do better. And for oncology, it's almost here. So there are better ways now to use genomics to understand the tumor behavior, right? There are better biomarkers, including liquid, which can be gotten from liquid biopsies, basically from the blood rather from the tissues. There are better tools for analyzing data sets, computational power, as I mentioned, using artificial intelligence, comparing it to what other people have seen. And then there's something called deep phenotyping, which gives you a lot more information than what we traditionally do, right? So this is the future, especially for blood cancers, for some solid tumors and so on. And then on the right side, you see what can be done. So you can actually subgroup, take material, learn about the biopsy, do drug screens ex vivo, get more information. And at the bottom, you can even see attempts to create xenografts. So you can take breast cancer tissue, and then in this case, we're looking at lung, and then model that as a xenograft in a mouse, and then use the mouse as a way to screen for potential therapies which are effective and less toxic. That field is almost there. Certainly at Johns Hopkins, this is done very routinely in our cancer center. But thereby you can come up with small molecules, many of which are available and can be repurposed, which are very effective and less toxic. And especially in the field of immunotherapy, you can develop specific antibodies towards certain tumors. So again, I'm not an oncologist, but in their field, it seems like they've made some headway. All right. Unfortunately, this potential for using genomics in cancer is not available to everybody. So this slide here is very instructive. So the area in blue shows where the field is moving. Clearly, countries in the global south are not in the picture. Africa is mostly empty, and Latin America. So this is an example of a huge global effort to look at as many as 2,600 different types of tumors and understand their characteristics and come up with specific therapies, and we are nowhere to be found. It's a problem, but it's also an opportunity. So that's, again, the take home from this slide. There are opportunities in our system to basically to discover what hasn't been discovered. Cancer drivers, issues about non-coding regions in the genome, mutational signatures of tumors, the natural history of cancer, evolution, RNA alterations, and so on, in order to be more precise in understanding tumor behavior and coming up with therapies. Okay, now to shift gears and come to diabetes. This is what I do for a living. In this country, in the United States, it's diabetes, endocrinology, and metabolism. It's actually 3 specialties in one. So diabetes is a very common disease. So worldwide, the prevalence is about 10%. In Ghana, I don't know what the precise numbers are, but it looks like about 5%. But Africa is going to see the largest explosion of diabetes in the next 20 years. The prediction is 134% increase. That's going to be a huge burden. Diabetes, as many of you know, is a major, major risk factor for eye disease. Every layer of the eye is affected. Kidney disease, cardiovascular risk, risk for infections, okay? You've seen that with COVID, non-alcoholic fatty liver disease, dementia, cancer, and so on, the list goes on. So we're basically sitting on the time bomb. It's a very expensive disease. In the United States, it accounts for about 1/4 of all healthcare expenditures. And then many patients, despite attempts to control their blood sugars, don't do very well. They still suffer end organ complications. So clearly there's a very unmet need for risk prediction for that. Who develops diabetes? And how can we specifically prevent diabetes or treat it in order to avoid complications? So this is the traditional way of looking at diabetes. Patient comes to you in Korle, you're likely, oh, this patient has type 1 or type 2. Trust me, there's a lot of hand waving. It's not as simple as you think. In order to make a diagnosis of type 1 diabetes, you often need to establish autoimmunity. Are you measuring antibodies? Which antibodies are you measuring? And what's the natural history of what you're seeing? Because type 1 diabetes is a disease where you have autoimmune mediated destruction of the beta cells which make insulin. So these patients ultimately have 0 insulin. They show up in the emergency room very sick, mostly with ketoacidosis. And if they don't get insulin, it's a death sentence, okay? Type 2 diabetes is actually multiple diseases. We often think, oh, this patient has type 2. My friends, you don't really know they have type 2 diabetes. All we know is they have chronic hyperglycemia and they are often obese, right? And we say they have type 2 diabetes. But we know groups of people who are predisposed. Family history is very important in type 2 diabetes. Aging, ethnicity, people of African ancestry are predisposed. Native populations in the United States, Native Americans, Pima Indians, and so on. Mexicans, Southeast Asians, South Asians, Pacific Islanders. Folks have a lot of predispositions towards type 2 diabetes. A hallmark of type 2 diabetes is insulin resistance. So these patients make a lot of insulin, they simply don't respond very well. Associated with a relative insulin deficiency. So over time, they simply don't make enough insulin to compensate for the degree of insulin resistance. And therefore they develop hyperglycemia. And the scary thing is that many of these patients are asymptomatic for years. So they may be going 10 years, 15 years without even knowing they have diabetes. And the first time they show up, maybe they have a retinopathy or they have nephropathy and so on. So again, getting to the fact that if there was a way to more precisely predict who's going to get it, we'll be in a better shape. And then there are rarer forms of diabetes which have to do with single gene mutations and chemical-induced diabetes and so on. Okay, so I mentioned type 1 diabetes. It's an old disease, and the slide I have on the left is a child. This is 100 years ago, right? Diabetes was a death sentence. This is how this child looked like, cachectic and shriveling to death, right? And insulin was discovered in the University of Toronto, quickly extracted from porcine islets and all later on from bovine islets. And you could see even way back in 1922, you could treat this child and she looks very good, right? Telling you insulin is a major anabolic hormone. It doesn't just regulate glucose, it regulates fat and protein. So the child quickly, and even water content. So the child fills up, she looks good. So this type 1 diabetes, we've come a long way. Now we use recombinant insulin, we use insulin analogs, and we've moved on from needles, which you see in the picture, into using insulin pens, which are more practical for most patients, into using insulin pumps. Most of them are open loop, and we're moving more towards the use of closed loop systems and so on. The detection systems for glucose have improved markedly. It's basically in people's pockets now, so they can use their cell phones or they can use their Apple Watches in a more productive way instead of just watching movies to be able to monitor their health and get better. So we've come a long way when it comes to type 1 diabetes. We still haven't treated autoimmunity, but at least we can replace the insulin and for most patients help them, right? Improve their overall health and reduce mortality. What about type 2 diabetes? I mentioned it's a very complex disease for which the etiology is multifactorial. It's also a multi-organ disease, as you can see in this graphic. So the hallmark of it is chronic elevation of blood glucose. It's hyperglycemia, but you can see several organs are affected. Patients have insulin resistance, so muscle and adipose tissue and liver don't respond very well. The net effect is the liver puts out too much glucose because you need insulin to drive glucose into the liver in order to make glycogen. If you don't have enough insulin, you're not responding well, you have excess hepatic glucose production. You have glycosuria. So you pour in glucose, you exceed the renal threshold for glucose, and glucose shows up in the urine. More recently, over the past decade, there's a huge interest in so-called incretin hormones. These are hormones which are made in the intestine, GLP-1 and GIP and so on. So these are very important. So patients with type 2 diabetes tend to be relatively deficient in GLP-1. And that's why we use GLP-1 agonists. And even the brain is tied into this system. I'm not going to go, that's what I spend most of my basic science doing, how the brain regulates glucose homeostasis. So in other words, the take-home message is type 2 diabetes is not one disease. It's a very complex, multi-systemic disease. Many organ systems are affected, and the end result is organ dysfunction, multi-organ dysfunction. Net effect is hyperglycemia and complications. And you see that reflected in the next slide. So a common dictum in medicine is if you're using too many medications, perhaps you don't have your diagnosis. And the answer here is very instructive. Why do we have so many medications for type 2 diabetes? Because we're not treating one disease, right? So some of your patients are on insulin. Again, hand waving, you don't even know they have insulin deficiency, do you? But we decide to give them insulin, and the net effect is many of them gain 10 to 20 pounds because we don't know what we're doing. Most patients are on metformin, which is an insulin sensitizer, works mostly in the liver to improve glucose handling by the liver, reduce hepatic glucose production. But we also have many other medications. We can flog the islets to make more insulin by giving sulfonylureas. That's an old-fashioned drug. More and more patients don't need sulfonylureas because there's huge risk for hypoglycemia. We can use glynides. There are amylin mimetics, which can improve insulin secretion. I mentioned the GLP-1 system. So there are GLP-1 agonists, which can be given. You can give DPP-4 inhibitors, which prolong the endogenous GLP-1 levels and improve glucose-mediated insulin secretion. So, and then we even have drugs which can cause glycosuria, SGLT2 inhibitors, which allow the kidney to pee out more glucose and lower the glucose. So the bottom line is that all these medications are available, but there's no precision as to who will benefit from what. So you run into the risk of reduced efficacy, side effects, and frankly, a lot of waste of money, right? Because we haven't precisely defined this disease. So this is one area where there's potential to use precision medicine to better characterize diabetes. The next slide shows you some tools that are used. So there's a genome-wide association. Basically, this is a descriptive marker of gene alleles, gene loci, which are associated with type 1 diabetes. Not to bore you, on the left, type 1, most of these genes have something to do with autoimmunity and inflammation. Not surprising, because it's an autoimmune disease. And so in the future, we might be able to use information like this to be able to predict which individuals and families may be at risk for type 1 diabetes. On the right, we have type 2 diabetes and the discovery of different loci over many, many years. What I want to point out from this picture is, you see the purple? These are all European populations, and maybe the black there is a few black populations. The take-home message is that so much discovery has been done in terms of gene loci associated with type 2 diabetes, but relatively little in people who look like you and I. So again, it's an opportunity to know more about type 2 diabetes, to discover more in our type of people, right? So most discoveries have been made in people of European ancestry. And as I'll point out later on, these are less diverse populations. The most genetic diversity is actually in Africa. We may look alike, but we are actually very different people compared to Europeans, where you have very few founders who moved from Africa there. And so their gene pool is actually less diverse compared to Africa. Okay, so this is my last slide, and it's going to be a lot of take-home messages. So we often get bogged down with challenges, and something's not going to work, something's not important. Unfortunately, in the field of precision medicine and genomic medicine, certainly from the perspective of Africa, most people think we'll be wasting money and so on. I think that is misinformation, okay? We don't think it's an area of priority. We have to worry about treating parasites and so on, okay? And then facilities simply are not there. And also, there are challenges to do with commercialization. Okay, people come there, take data, and you don't even know where the data goes, right? They go to their populations and enrich themselves at our expense. On the right side, I think opportunities. There are always opportunities in the midst of what we think are problems. We have a huge untapped resource to actually redefine diseases from our perspective and actually to inform the world, okay? Because after all, humans came from Africa. We have opportunities to use the tools I mentioned to understand more about human genetic diversity. Where did we come from? How did we evolve the way we did? What is disease causation? Why is the pathogenesis of COVID in Africa seemingly different from other parts of the world? So there are hidden germs there which we can really tap into. Not only with humans, our environment is actually more biodiverse than any other continent. So there's a huge opportunity for us to use genomic tools and other biomarker tools to explore this area. I mentioned the natural history of diseases. So talk about cancer, talk about diabetes. Any disease you can think about, this is our opportunity to redefine diseases. Okay, people talk about costs, and I threw in some number here. When the human genome was first sequenced in 2000, 2001, it cost the US taxpayer $100 million to sequence just one person. Guess how much the cost is? Actually less than $1,000 in 20 years later. It's very cheap now. It's basically in your back pocket for most people, and most of that 1,000 is actually labor costs. So I can guarantee you can sequence somebody's genome in Ghana for as little as $200. It's possible now, okay? So the technology is more affordable and accessible, and we can hand wave and talk about, think about how many people you can sequence for the cost of one SUV, right? Which people drive around in Ghana. And think about the benefit, the downstream benefits of that, okay? It can open up opportunities for the development of biotechnology unique to our environment and actually teach the world something. Opportunities for informatics, for computational biology, obviously huge health benefits, right? and overall socioeconomic benefits. Is this being done? I've just thrown in an example here. So I have a colleague and friend who some of you may know, Charles Ahima, is a professor in the University of Amsterdam. He's a PhD. He has a very interesting cohort where he's studying Ghanaians who live in the rural or urban areas and matching them with people who look like them, come from the same ethnic groups and live in Europe. It's called a RODAM study. And the focus is on diabetes, obesity, cardiovascular risk, and so on. Very interesting cohort. So Charles reached out to me maybe 5 years ago. We've become very close friends and we've been collaborating. He has about, I think, close to 7,000 people in a cohort now. So very unique set. And we're hoping to mine this data and understand more the genetic diversity, what the genome has to do in relation to diabetes, cardiovascular risk, and very importantly, the impact of environment. Remember I mentioned rural, urban, and migration. This is a very unique data set that we're trying to build on. Hopefully we'll be able to tell you more down the line. On the right side, I have other attempts at doing genomic work in Africa, funded mainly by the Wellcome Foundation in the UK, and also H3, which is funded by the National Institutes of Health. The emphasis is here on diabetes, cardiovascular diseases, kidney disease, cancer, sickle cell, and so on. So we are not in the dark completely. To me, the take home here has to be, we have a huge opportunity to do better. And if we play our cards right, there's no reason why Africa should not be the beacon in the fields of personalized or precision medicine and genomics. I think that's my last slide. Thank you.